Hello everyone, I'm Dr. Arpita. The topic for today's discussion is glyoxalate cycle. Glyoxalate cycle is an anabolic metabolic pathway occurring in plants and several microorganisms like bacteria and yeast. The glyoxalate cycle centers on the conversion of acetyl coenzyme A to succinate for the synthesis of carbohydrates. It is absent in animals including humans. Its absence explains why humans can synthesize glucose from pyruvate and TCA cycle intermediates but not from acetyl coenzyme A. So this is the pathway that shows what I have just told you. Because animals do not run the glyoxalate cycle, they cannot produce glucose from acetyl-CoA in net amounts, but plants and bacteria can. As a result, these organisms can turn acetyl-CoA from fat into glucose, while animals can't. Bypassing the decarboxylations and substrate level phosphorylation has its costs, however. Each turn of the glyoxalate cycle produces one FADH and one NADH instead of the three NADHs, one FADH2 and one GTP made in each turn of the citric acid cycle. There are no decarboxylation reactions in the glyoxalate cycle. The glyoxalate cycle allows cells to utilize 2 carbon units of acetate and convert them into 4 carbon units succinate for energy production and biosynthesis. Additionally, each turn of the cycle produces a molecule of FADH2 and NADH. The two key enzymes involved in the glyoxalate cycle produce 2 malate molecules, which in turn produce 2 oxalate molecules. The spare oxalate molecule is used in gluconeogenesis for glucose production. As we all know, gluconeogenesis is the synthesis or generation of glucose from non-carbohydrate sources like proteins and fats, and this mainly happens in liver and kidney, and it happens when the supply of carbohydrate is low, that is when we are starving or when we are on low carb diet. So coming to the two enzymes here, isocitrate lyase and malate synthase. Isocitrate lyase converts isocitrate containing 6 carbon to succinate containing 4 carbons and glyoxalate. Malate synthase combines acetyl-CoA and glyoxalate to yield malate. Now we'll discuss the steps. The pathway begins with 2 molecules of acetyl-CoA. This is followed by citrate synthase that converts one of the acetyl-CoA molecules to citrate. Citrate is then converted to isocitrate by the enzyme aconitase, and isocitrate is converted to glyoxalate and succinate. Succinate is then converted to fumarate by succinate dehydrogenase. The next step involves the formation of 2 molecules of malate. One molecule of malate is formed by the combination of acetyl-CoA and glyoxalate, and the second molecule is formed by the conversion of fumarate to malate in the presence of fumarase. Then malate dehydrogenase converts 2 malate molecules into 2 oxalate molecules, and 1 molecule of oxaloacetate is converted to citrate, and 1 molecule of oxaloacetate is used for gluconeogenesis. So in the last 2 steps, what we can see, the malate dehydrogenase is converting 2 malate molecules into 2 oxaloacetate molecules, and one of these is converted to citrate and the other is used for gluconeogenesis. So this is a pictorial representation of what we have just discussed. You need to follow each of these steps in order, and your answer should be accompanied by a diagram. So this is how we come to an end of our brief lecture on glyoxalate cycle. If you have any confusion, any doubts, please feel free to get back to me. Till then, thank you and bye-bye.